All right. I also talk about active transport in zebrafish, which we heard already a bit today from Michael Gottesman. And I'll talk about zebrafish embryos. And I'm at the Helmholtz Center for Environmental Research, so I'm not really in medical research. It's really more environmental research. That's the background of my work or our work. So we're interested in ecotoxicological effects of environmental chemicals and how these effects can be used to assess environmental risks. And well, actually you're actually required to do for tests of chemicals with adult fish, but these are animal experiments, and this raises the questions, why not use fish embryos as an alternative? Because they are not considered as animal experiments. They are faster, cheaper, and there are lots of diagnostic options in the embryo. But well, you need data for adult fish, actually, for chemical registration. And when you compare the toxicity of chemicals to embryos and adult fish, you have a correlation, but you have a lot of outliers also. So it's a question if you can make a good, let's say, prediction of toxicity for adult fish from fish embryos. And therefore is the question, how can fish embryo toxicity really serve to predict adult fish? So we need to understand why chemicals are toxic to adult fish and why they are toxic to fish embryos, so we can kind of make a prediction for adult fish, basically to not do so many animal experiments. And as you can see, I mean, fish and adult fish and fish embryos are quite different. You can see it already here. And the embryo is a very dynamic system. So this is a zebrafish embryo. Within 3 days, it develops from a single cell to a complete organism. And it doesn't take up any food. So during development, so in these 3-4 days, first 10 days, it actually doesn't eat. So it loses a lot of weight. Body mass parameters decrease. So you see here dry weight, carbon, nitrogen. And also gene expression is extremely dynamic. So this is transporter protein that we heard already today about, and which I will also talk about. It's zebrafish ABCB4, and you can see that is the constitutive expression of this transporter in the first, let's say, 5 or 6 days, I think. And these data are actually from omics data, so microarray and sequencing data. And Paul Michaelis, who works in our lab, he kind of analyzed the control data from experiments, the control data to look for constitutive expression of this transporter gene. So basically, we try to understand the parameters that determine toxicokinetics. So it's equivalent to pharmacokinetics of chemicals in the fish embryo. And so this again is ABCB4. When you knock it down, so we found when you knock down ABCB4, zebrafish embryos, so these are these arrows, they become more sensitive to the exposure to vinblastine. So vinblastine is a known cancer drug, and ABCB4 actually keeps it out of the fish embryos. So it becomes more sensitive when the transporter is knocked out or inhibited by a chemical. And accordingly, it also keeps out dyes, so calcium AM, for instance, or rhodamine, and we use that to look for the to make or perform kind of a dye uptake assay with fish embryo, with live fish embryo, using the so-called VAS system. So this system is an automatic, semi-automatic system. It drags the embryo into a capillary and positions it in front of a lens of a microscope, and then fluorescence photographs can be taken from the embryo. And as you can see here, so when you don't add any inhibitor, the embryo is quite dim. But when you have an inhibitor of the transporter present, of this efflux transporter, the embryo appears much brighter, and you can quantify this signal, yeah, and then quantify kind of the effect of the inhibitor on the efflux transport. And in parallel, we performed ATPase assays with recombinant zebrafish ABCB4 protein. And well, the principle is that when a compound interacts with a transporter, ATPase activity is stimulated and phosphate is released from ATP cleavage, and you can quantify that, measure it. And we looked for correlation. So we tested a number of chemicals and looked for correlation of the results. So we performed single protein ATPase assays and whole embryo dye accumulation assays on the other side. And as you can see, there is a correlation of the data, but there are lots of outliers, and we think actually that this is a sign that there are other transporters in the zebrafish that also are responsible for efflux of rhodamine dye. And so other transporters are active. And we looked for another transporter when we had actually this data. So this is an image of zebrafish embryo, and it was exposed to the compound bromoxynil. So bromoxynil is a herbicide containing brome, and you can actually visualize the brome. So on the left, you see a laser ablation MS image, and it kind of detects the brome in the bromoxynil. And you can see here the a signal in the, it's kind of in the middle of the embryo, so probably the intestinal tract. And a similar image we got with the multi-MS image, where you also actually see the signal here in the middle. So it's probably the gastrointestinal tract. And the gastrointestinal tract expresses a certain protein, OATP1D1 is called. It's an SLC transporter that we studied, and it's OATP1D1 is an uptake transporter, and cells were transfected with this transporter. And when they were exposed to BSP, the ones that were transfected, so overexpressed OATP1D1, took up much more of the BSP than the control cells, which is kind of expected. It's an uptake transporter. But when the cells were exposed to bromoxynil, it was quite an interesting result, because there was less accumulation of bromoxynil in these cells than in the control cells. So, which is an indication that this transporter, which is supposed to be an uptake transporter, also has an efflux function. Yeah, and this, you know, when we got this picture, we thought, okay, it makes sense. It's an uptake transporter, so probably it accumulates. So the cells of the gastrointestinal tract accumulate more bromoxynil, and therefore we get the signal. But when you think about it, okay, when it's an efflux transporter, it may also make sense because it effluxes the bromoxynil into the lumen of the gastrointestinal tract, which might happen, yeah. So, okay, so basically the take-home messages, so transporters are important, toxicokinetic relevant parameters in the zebrafish embryo, and we want to right now look at more ADME related enzymes. So we go to human and look which pharmacokinetic or enzymes do we find, look for more candidates in the zebrafish. Are they expressed? What are the functional properties? And how are they involved in toxicokinetics? So the work that I showed was basically done by many people. So these were main people like Stefan. He did a lot of the ABCB4 work. Katharina did the OATP1D1 research. Flavia actually worked with the VAST system, and Kathleen did all the ATPase assays. And these were the funding sources. Thanks very much for your attention.
And thank you for a very interesting presentation, Sil, and for sticking to the time. So, yeah. I mean, I have one question. So how many transporters are expressed in the zebrafish? Because you have all these gene expression data you mentioned, both RNA-seq and microarray.
Yeah, actually, you know, Paul is just doing the calculations, and I sent him a few. I really don't know how many, but it's quite a number. So he looked for different ABC transporters, and a lot of those, so these MDR transporters, so also ABCC, 1 till 6, I think, he looked at, and they pretty much all expressed. It's just you have to, because I said the expression is quite dynamic, so we have to really look at which stages. So at a certain point, they are all expressed, but in the very early stages, expression is quite low. So, but yeah, good question. But we really have to look further into that, what the expression patterns are.
Do you plan to knock them out like one after one, or what is your plan to dissect it? Yeah.
That would be very great, yes. Yeah, I'm just starting this. So I did, Stefan was a PhD student many years ago, and I didn't do a lot of research now, but plan to really kick off now with zebrafish and do more work and use the CRISPR-Cas maybe for knockout and all that. So, but we haven't started yet right now.
Any other questions? Anna?
Yes, I would like to ask a question. We have seen that many detergents are substrates or alloprides for transporters, and at higher concentrations they also inhibit the transporters. And now I wonder, I mean, we let a lot of detergents, tons of detergents per year into the water, and I'm always very much afraid that, you know, we come to a point where the water is so contaminated that these transporters may not defend their beings, the living, the fish and anything, the embryos particularly anymore. And do you see something, I mean, do you also feel that this is a danger, or what is your feeling about detergents?
Yeah, so it's always a question if those compounds reach the transporters. Yeah, so it's a matter of uptake and where they end up. So it's actually hard to say. So I tell you right at the beginning, I really don't know, but there is definitely concern. And also because there are many compounds. So as we heard already, now the transporters are multi-specific, and and we have thousands of compounds in the environment, and even if they are at lower concentrations, together they can act, in a combined way. So that is really a concern, and something I would like to address more. Yeah, because we have also some evidence that certain compounds, they act as inhibitors, as you mentioned, for detergents, but also other compounds like fragrances or certain pesticides also.
Anything which is amphiphilic, yeah.
Yeah.
Probably.
And yeah. So that is definitely concern we also have, that could be a significant effect. Yeah.
And the point is, even if they are not toxic, so that's the, detergents are per se not toxic at low concentrations at least, and but still they do the effect together with others.
Exactly. So
thank you very much.
Oh, it's a valid point. Absolutely.
To add, you're right. So they are not toxic, and so they are not considered as toxicologically relevant. But this effect as, let's say, chemosensitizer is not considered, at all in regulation. Yeah. Mm-hmm.
Okay. Thanks.